So in this finding one nucleotide chain is running in 3 prime 5 prime direction, the other is running in 5 prime 3 prime direction, and in the backbone we have sugar molecules and phosphate, and in between we have this nitrogen base pairing. So adenine with thymine has double bonds, guanine with cytosine has triple bonds. So between these two coils, the area is 3.4 nanometers. Each coil between the two nitrogen bases, we have 0.34 nanometers. So we have major groove, minor groove. Just looking at this picture, you can just write Watson and Crick's model better, very, very easy. So this picture is also same thing, Watson Crick model only. And the diameter of this coil is 2 nanometers. So again here ATGC is shown and phosphate is shown in zigzag way, and you have like sugar as backbone. Okay. So phosphate. So one end is 5 prime, the other end is 3. At 3 prime end we have OH group. Okay. So just that is Watson Crick model, but it's not so great thing. It's easy only. If you read once, you will really understand. Proteins based on the functions, he said, right? So for that, we have based on the functions what they perform, we have structural proteins like keratin of hair and nails, collagen of bone. Then enzymes are catalytic proteins. We have hexokinase, pepsin, transport protein, hemoglobin, serum albumin. Hormonal proteins, insulin, growth hormone, contractile proteins, what we found in muscle fiber, actin, myosin, storage protein, ovalbumin, glutein. Then we have genetic proteins, nucleoproteins, nothing but DNA, RNA. Defense proteins like snake venoms, immunoglobulins, receptor proteins for hormones and viruses. Okay, so all these are, now he is asking us to elaborate on hemoglobin and collagen. Collagen structure we did recently, and hemoglobin, the structure of hemoglobin also I told you it has 2 alpha chains and 2 beta chains. That particular structure you have to focus, like elaborate more when you get this question. Okay. Then tumor markers also we did. Anybody remembers few examples? CEA, carcinoembryonic antigen, isn't it?
Fetal,
The
fetal.
potassium levels you can check from cover page, beta. Hyperkalemia, what are the diseases? Niharika, can you share on the screen? Potassium.
Yes, ma'am.
Potassium.
Potassium.
Under diseases of potassium, below functions you will get hypokalemia, hyperkalemia. Hypo means low potassium levels, hyper means more potassium levels. Detoxification we did, isn't it? We have hydrolysis, we have conjugation, oxidation, reduction, everything. Type 1 and type 2 reactions. Types of blotting techniques. Okay. Oh, body's immune response to coronavirus infection. So bodies, because it is virus, no? So what type of immunity will be functioning in case of viral infection? Cell-mediated immune response. Okay. So in cell-mediated immune response, in the sense, T lymphocytes will be functioning. Okay, so under T lymphocytes, you will see many inflammatory mediators will be released like histamine, serotonin, bradykinin, tumor necrosis factors. All T cell mediated immune response will be there in case of coronavirus infection. So not only corona, any virus infection, usually T cell mediated immunity works, not B cell. Okay. So you have to talk about T cell mediated immune response for this answer. So blotting techniques I will check. Meanwhile, you share this potassium. Okay.
Yes, ma'am.
Blotting techniques. Yeah, very good. Beta. Thank you. So hypokalemia. So reference range. Reference range also you can check from the last page. And what happens if there is, okay, hyper, both are asked or high. Okay. Hyperkalemia, increase in the concentration of serum potassium. So when do we see this? In renal failure, adrenocortical insufficiency. That means aldosterone deficiency that we didn't study it even in physiology. That is Addison's disease. diabetic coma, severe dehydration. You can give few examples, whatever have been discussed here. Okay, that will lead to hyperkalemia. Hypo means low, Cushing syndrome, okay, and cardiac arrest, muscle weakness, and all. Hyper is more potassium levels in the blood. Emia is always blood. Okay, so let me check with blotting techniques. Types of blotting techniques. So there is a question like blotting techniques for you, isn't it? So this blotting techniques, it has come for how many marks? So for 3 marks, no? So types of law along with its clinical applications. Okay. So first of all, you have to give the list of this better. Like we have blotting techniques are southern blotting technique for DNA, northern blotting technique for RNA, and dot blotting technique, which is used to check for DNA and RNA. Okay. So these blotting techniques are generally used to detect, suppose for medico legal importance will be there, no, like in forensic medicine, you get a piece of hair, you get a piece of nail, or you get some blood stain or something, with the help of which you will just detect like who is the criminal by just checking like what are the real genes in that particular person and if they are matching or not. Generally, these blotting techniques are generally done to detect. See, like the blotting techniques involves the transfer of protein blots and their identification by using specific antibodies. Already existing antibodies of that particular person will be matched. So we can detect DNA, RNA, DNA and RNA together. So he is first, once you give the list, now he is asking uses. Forensically, I said no better, forensically applied to detect minute quantities of DNA, to identify parenthood, to identify thieves, rapists, etc. Okay, generally we make use of this southern blotting technique. Okay, so not the because southern blotting technique is for what are we doing it for? DNA, isn't it? Now let us see what are the uses of northern blotting technique. Is a good technique in determining number of genes which are present in mRNA. That means what may be the size of protein which can be synthesized with the help of this mRNA. We are determining that with the help of this northern blotting technique. Then dot blotting is particularly useful in obtaining quantitative data for the evaluation of gene expression. Okay, so these three uses you can write. Okay, so give this and write the uses. Okay, beta, so we will stop here. See you tomorrow, same timings. Sunday I may change, but tomorrow same timings. Okay, beta. Good night, all.